children look different from their parents, and so do the young ones of other living organisms. What are the reasons for such variations? To find the answer to this question, Thomas Hunt Morgan and his colleagues tried to verify the chromosomal theory of inheritance through various experiments. Morgan chose the tiny fruit fly Drosophila melanogaster for his research. There were several reasons for this. First, these flies could be easily grown in a simple synthetic medium in a laboratory. Morgan also found that a single mating in these flies produced a large number of offspring. Moreover, the male and the female flies were easily distinguishable and had only 4 pairs of chromosomes, making their analysis easier. Another reason for Morgan selecting these flies was that they showed several heredity variations which could be easily seen through low-power microscopes. Finally, these flies had a life cycle of just 2 weeks, which would help study several generations within a short span of time. Morgan carried out several dihybrid cross experiments with Drosophila to study sex-linked genes. For example, he crossed yellow-bodied, white-eyed females with the genotype y/w with brown-bodied, red-eyed males with the genotype y+ and w+ to produce the F1 generation. Here, the plus sign in the superscript represents the dominant wild-type alleles. Morgan then intercrossed the F1 generation to obtain the F2 generation, and observed that the genes did not separate independently, which was an exception to Mendel's law of independent assortment. Morgan and his colleagues knew that only the X chromosome bear genes. Furthermore, they noticed that during a dihybrid cross, if 2 genes lie on the same chromosome, then the proportion of parental gene combinations, that is, y/w and y+w+ in this case, was much higher than non-parental gene combinations or the recombinant types, that is, y+w and y/w+. Morgan found that this was due to physical association or linkage, which describes the tendency of certain genes to be inherited together and their ability to retain their physical combination in progeny. Genes which are located on the same chromosome and that are inherited together are known as linked genes. For example, it is the linked genes that account for a phenomenon like red hair being strongly associated with light-colored skin in human beings. If an individual inherits one of these traits, he or she is most likely to inherit the other trait too. This phenomenon of linkage can be better understood through a process known as crossing over, which occurs during meiosis. During the Pachytene stage of meiosis I, sections of a chromosome usually intertwine and exchange genetic material between non-sister chromatids of homologous chromosomes. This process, where chromatids of a homologous pair exchange segments between them, is known as crossing over. Crossing over leads to recombination or the creation of different combinations of alleles that do not exist in either parents. And hence, it is one of the most important events that lead to variations in offspring. Another fact discovered by Morgan and his colleagues was that the likelihood of 2 genes separating during crossing over was proportional to the distance between them. In other words, the closer the genes, the more likely that they will stay together. And the further they are, the more likely that they will separate. Now, let's consider another experiment conducted by Morgan, where he crossed white-eyed miniature winged females with the genotype w with red-eyed normal winged males with the genotype w plus m plus. From his experiments, Morgan noticed that tightly linked genes, or genes nearer to each other, showed very little recombination, while loosely linked genes, or genes far from each other, showed higher recombination. For example, the yellow and white genes showed a recombination of just 1.3%, as these genes were tightly linked, while the white and miniature genes showed a recombination of 37.2%, as these genes were loosely linked. This linkage and the frequency of recombination of genes on the same chromosome were used by Morgan's student, Alfred Sturtevant, as a measure of the distance between genes to map their position on the chromosome. For instance, in the experiment we just saw, it is only through mapping that we learnt that genes y and w were located close to each other, and genes y and m were farther apart. Today, genetic maps are being extensively used for linkage studies and analysis, and are a key tool for genome sequencing or discovering the location and identity of genes. Thus, Morgan's experiments on linkage ruled out Mendel's law of independent assortment when linked genes are considered.